from evolving dragonflies to invading lanternflies, how your observations can lead to amazing discoveries. It features our speakers, who I think you can see in the top here, Dr. Carrie Seltzer and Dr. Michael Moore, as well as your observations. And of course, meeting digitally is not our usual method, but it does allow us to include a wide range of audience members from across the country. In fact, I know this evening we've already got people logged in from Florida to New York, one in Oregon, some in California, and a wide range of locations in between. In fact, our speakers, Carrie is with us from Maryland and Michael is with us from Missouri. And our audience also includes family members of the Robert Hefner family. We have Miami professors and alums from many generations. We also have students logged in tonight. I can see a lot of them in, and I know that some of them are only here for the extra credit, but that's okay. I'm sure it'll be fun too. And of course, we have friends of the Hefner Museum from all ages and disciplines. Now, we are recording the event tonight, and we have set up the broadcast, though, and the recording so that only the panelists' images and voices will be recorded. So to be clear, participants will not be recorded. If you're viewing, no worries, we can't see you, and you will not be part of the recording. However, one positive aspect of this digital format is that you viewers can submit your questions during the presentation, and as appropriate, I may voice your questions to our speakers, and if it's not appropriate to voice the question to the speakers immediately, we may answer that at the end of the talk, or I may even answer them synchronously in the chat. I see that we have somebody from Texas just logged in. Welcome, Sam. You can also submit your questions through Zoom chat, which is here, the Facebook or Instagram, and we're also watching our YouTube live comment section too. We've got a wonderful intern on the other end of the gallery here who is monitoring our chats there. So please ask away if you have any questions. We did learn a few hours ago that closed captioning services are offline right now, so we are going to work quickly to post a closed caption version of this talk in the next few days. Now, given the digital format of tonight's lecture, we're going to forego a handful of our lecture traditions, including the service awards. And of course, we're not going to be able to have those wonderful stuffed mushrooms at the post-lecture reception, unless you've got some with you right now. But I will hang around after the lecture so that I can take my laptop around the museum and show you all the fun things that are going on if there are any questions about that. But before we begin with the lecture by Carrie and Michael, I want to take just a moment to thank all of our supporters. And I'm going to just pop up a little slideshow here for a moment as I thank folks. I'm going to share that screen. All right. So, of course, I want to thank our student and community volunteers. We work with about a dozen majors each semester, and they contribute their times and talents and oftentimes learn a thing or two as we accomplish our daily work here. We're also exceptionally thankful for our community volunteers because we can rely on those folks almost any time of day, year round, to help with some of our most complex tasks. And indeed, as we've been working through quarantine conditions here, we've worked with community volunteers, including John Kinney, on many of our projects, such as the Imaginarium, which perhaps you saw in the earlier slideshow. I also want to thank our corporate foundations, our corporate and foundation sponsors, because they're really key to both maintenance of the museum and our innovative projects. I want to recognize Project Dragonfly is here tonight with us, helping us with the tech. The OEEF, the Ohio Environmental Education Fund, is funding much of our renewal and refocus of the Imaginarium. The Oxford Community Foundation has contributed funds to allow us to adapt exhibits to people of all abilities. The Bever Family Foundation continues to support many of our staff and innovative work, as does Duke Energy. And we couldn't do all of the staff work we do without these foundations. And the One America matching donations really help with a lot of our exhibits, especially Cat Mountain. We also want to thank all of the many friends of the Hefner Museum. We have a variety of donors. Bill and Ellen Cam, One America, and Sally Sue Siebold have contributed substantial amounts to some of our exhibits, especially to the Cat Mountain, which has really helped bring that exhibit up on our priority list and really get moving on that. And it's going to be an exciting exhibit. We have tributary donors who we can always rely on to provide funds for us throughout the year. And in fact, at only the cost of one coffee a day or one coffee a week, you can join the tributary donors. And the charismatic species donors, we have lots of charismatic people, not just in name, but I love hanging out with all of our friends because they are so charismatic and teach me so many amazing things. And I want to also acknowledge the steady support that we have from departments and local organizations that regularly co-sponsor the Hefner Lecture. The University Special Events Fund, Audubon Miami Valley, the Graduate School University Honors Program, and the Institute for Environmental Sustainability, and especially the Biology Department here in Oxford and the Regional Biological Sciences. We partner with them basically daily in all the work we do, as well as Project Dragonfly, the Geology, Environmental Sciences, Microbiology, Geography, and Kinesiology and Health. And we work with a lot of their students and benefit from a lot of the volunteers that come from those departments. And of course, we have to all thank Julie and Carla. If we can give all of them a big hand, silent as it may be in this format. Quite literally, tonight wouldn't happen, and all of our various activities wouldn't happen with these two wonderful staff members of the museum. So without further ado, I'd like to stop sharing here for a second and introduce our speakers. Maybe we can pin Carrie up while I introduce her. Dr. Carrie Seltzer is an ecologist who has worked at nature centers and the National Science Foundation with organisms from pouched rats to bats. And today she helps people turn simple cell phone photos into scientific data that records the world's biodiversity through the iNaturalist app that many of you have used and benefited from. And if you haven't used it yet, give it a try. I am arguably one of the more technophobic people in the world, and I have loved the iNaturalist app, and especially the artificial intelligence behind it. I literally learn something every day as a result of using this app. As iNaturalist's stakeholder for engagement strategy, Dr. Seltzer facilitates partnerships that expand the reach and use of iNaturalist beyond just you and me to scientists around the world to help us study the nature of your neighborhood and your backyard and beyond. Now, Dr. Seltzer and I met as students at the University of Illinois at Chicago. So she and I have looked at a lot of nature together. And according to Dr. Seltzer, she has used the iNaturalist app to catalog over 400 species in her yard alone. Now, she doesn't know this, but in the month that I've been using the iNaturalist app, I've been in a race with her. And since she doesn't know it, I've been able to catch up with her pretty quickly. And one of the amazing things, I think, is go check out my profile on iNat. I am approaching 300 species in my backyard in just a single month of getting up and taking breaks from quarantine computer work and walking out and finding something new. So I really can't wait to use this for an entire year and see how many thousands of species inhabit my suburban Ohio yard. And I encourage you to do the same thing. Now we switch over to Dr. Michael P. Moore for a moment here. Dr. Moore studies adaptation and diversification of life histories and mating systems, the various trade-offs that occur throughout an organism's life from the juvenile form to adult. So I like to think of this as like, think about a tadpole. It goes from little gelatinous egg to this little gooey fish thing to this little hoppy frog thing, and think of all of the different life stages that that organism goes through, and the conflicting things that go on. And that's kind of what's in my head when I hear Dr. Moore describe the kinds of trade-offs that he studies. Dr. Moore is presently the Biodiversity Post-doctoral Fellow with the Living Earth Collaborative at Washington University in St. Louis, Missouri. He earned his PhD at the nearby Case Western University, and he works with students of all ages, which makes him a real friend of the Hefner, because of course we love to work with people of all ages too. And he uses the data collected by folks like you through things like iNaturalist to make the amazing discoveries that he's going to talk about tonight. Now, Dr. Moore likes to joke that because he has a fear of both snakes and spiders, he only studies organisms with between 1 and 7 legs. So those of you that know me know that I'm going to try to convert Dr. Moore to the clan of ophidiophiles so that he can also enjoy snakes. And one day when quarantine's over, I'll throw a snake on your neck there so that you can have the benefit of the best massage ever. And of course, finally, because tonight's theme incorporates citizen science, I want to recognize all of you as contributors to tonight's talk. And both Dr. Seltzer and Dr. Moore will elaborate on how all of you have contributed so substantially, not just to tonight's talk, but to the better understanding of life on this planet. So without further ado, I want to turn the time over to Dr. Carrie Seltzer from iNaturalist.
Thanks so much for the introduction, Steve. I wanted to share that it's a real honor to be invited to give this talk, and I have a couple of connections to Miami University and to smaller natural history museums and colleges and universities. I went to Earlham College, which is just down the road from Miami University, across the border in Indiana, which also has a small natural history museum, the Joseph Moore Museum. And so I personally know the value of a natural history museum on campus as part of an undergraduate education. Steve and I met in grad school in Chicago, like he mentioned, but I've also got strong connections to the sort of Southwest Ohio area because my parents live in Dayton. So I was there at the beginning of September. I wish that I was on campus in person in Oxford to give this lecture, but maybe one day in the after times. Anyways, it's my great pleasure to share about iNaturalist. So iNaturalist is a platform for sharing your encounters with biodiversity and crowdsourcing observations. It exists to help people explore biodiversity through connecting with others. And I'll emphasize throughout how important people are in this. It's not just about data. So what this might look like is you're out, you see a cool butterfly land on a flower, you take a picture with your phone, you upload it to iNaturalist, and then even if you don't know what you saw, other people can chime in and help advance the identification. And collectively, all of these observations that we make all over the world can contribute to some exciting discoveries that we'll touch on. Every observation is really an invitation to connect with other people through iNaturalist about what you saw. I want to emphasize that iNaturalist is a platform that works across all your devices. You can use it on the website. You can use it on the free mobile apps on a tablet. The website does have the most functionality. So a lot of the screenshots that I'm going to show tonight are from the web. And if you haven't checked that out, if you've only used the app, I definitely encourage you to check out the website as well. I always like to thank the iNaturalist community at the beginning of my talks because it's just so important to recognize that iNaturalist wouldn't work without all of the enthusiastic people using it who share their expertise and give us an insight into what it is that they encounter every day. So thank you. And thank you to all of you who in advance of this joined iNaturalist and started adding observations as well. We talk a lot about observations on iNaturalist. So this is sort of the unit of interaction of most interactions on iNaturalist. And an observation captures some crucial pieces of data. Who you are, you can have a pseudonym, but everyone does have to have an account on iNaturalist to submit something. We need to know where it was that you saw it. We need to have the coordinates. We need some evidence of what you saw if you want other people to chime in. Normally that's photos, but it can also be sound. We need to know when you saw it, at the very least the date, if not also the exact time. And then there's this piece of what you saw, but you don't have to know exactly what you saw because other people can help with that. And even if you do know, other people can help confirm it. So all of the observations on iNaturalist, ideally, eventually, are connected to a taxon on this single tree of life. So what the cool piece about iNaturalist, I think, is that because there's this underlying taxonomy that unites everything, it means that even if you don't know exactly what you saw, adding a general identification like birds or plants, it is really helpful, because then it helps the people who are looking to help other people identify birds or plants to find your observation. If it sits in the bin of unknown observations, then someone may come along and add a very general identification in the hopes that someone with more expertise will then be able to find it. So if that's happened to you, you've been like, why did this person just say this is a mushroom? That's not very helpful. I can see it's a mushroom, but what is it? What is it? It's because of that sort of progressive process of identification. And through all of the tens of millions of photos that have been shared with iNaturalist, and the even more tens of millions of identifications that have been associated with those photos through the observations, we've amassed an enormous data set of generally well-labeled data of biodiversity from all across the world. And using these photos and the identifications, we've been able to train a computer vision model to suggest what it is you might be seeing. Steve mentioned this in the introduction. He's found it super cool. If you haven't tried it yet, I definitely recommend it. I would say in most parts of the United States now, it works pretty well, and it feels like magic when it works really well. Sometimes when it doesn't work, it's pretty hilarious, but there's usually a reason. If you get some really crazy answer, there's a reason. There's a reason for that. So here's just a glimpse of what it looks like with a screenshot from my iPhone. I took a picture of this violet in my neighborhood, and you can see that it gave me both genus level identification at the top, but then it also gave some species level suggestions. If any of you are familiar with violets, they can be pretty tricky. And so sometimes it's always important to click that little eye on the right side there to get more information. Look and see, is this something that's been seen nearby? Look at more of the photos, try to get a sense of whether or not it seems like a good match. And if you look at those suggestions and none of them seem quite right, perfectly acceptable to go for that broader one, or even to go all the way back to a much more general identification and just type in plants and say it is something in this kingdom. That's totally fine. We encourage everyone to sort of use these suggestions as a starting place, but take them as a grain of salt. Take them with a grain of salt. Don't take them as gospel or the ultimate answer. It's a starting place. So here's a heat map showing the parts of the world that have the most data. And right now, in terms of the amount of data we're talking about, we've got 52 million observations, and those currently represent at least 300,000 species, which is still just a fraction of the named species in the world. We've got a long way to go. You can see there are still many parts of the world where there's not a lot of iNaturalist activity, and many more species to be shared on iNaturalist. One of the ways in which you can aggregate observations on iNaturalist around a theme is using a project, and that's what was done in preparation for this lecture. If you haven't yet, I encourage you to join the Hefner Museum Nature in Your Neighborhood project on iNaturalist. And when you join this project, it's set up so that all of your observations for the specified time period will be automatically included. So thank you to all of the 75 people who have done this. I know Steve's observations are in there. Some of mine are, including some from the Dayton area. And this is just a neat way to see who else on iNaturalist has a connection to the Hefner Museum. And the observations in this project have come from a wide range of places. Most of them are from the US, but there are a few from China, which is super cool. Most of them from the Eastern US, but some from the West as well. And I'll highlight a few of the interesting ones that caught my eye. I also wanted to give a shout out to a couple of other projects associated with courses this fall. The invertebrate zoology course has had a lot of activity. They've actually got the 2 sections, it looks like, in separate projects. So if you want to see more about what they've found, look for the Miami U Invertebrate Zoology project. And then there's this 115, the Birds and the Bees project, that has almost 600 people. I hope that a lot of the students who are in this project are watching. This is super cool. I've enjoyed looking at your observations as well. And kudos to the professor. It seems like you've gotten some good direction on this. So those are just a couple snapshots of a few of the ways that you can look at data in aggregate on iNaturalist. But now I want to highlight just a couple of cool observations that caught my eye. So this is an American martin that was spotted out in New Mexico. And what I think is cool about this is that, first of all, it's not super easy to see or photograph a martin. And so these are some pretty awesome photos. So good job, Bilo Bree, for taking these photos and posting them to iNaturalist. If you look at the map too, you can see there's not any orange dots around that one, which means that no one else has posted an American martin observation from that view of the map yet on iNaturalist. So you're sort of helping fill in a gap in the map of iNaturalist data about where American martins occur. I thought this was pretty cool. Wee Corby has submitted tons of observations. I hope you're watching, Wee Corby. And this one caught my eye because it shows a rat snake climbing brick, which is just so cool. They're such great climbers. This was observed down near Cincinnati. The location has been obscured. I do that for observations from my home to protect my personal privacy, but we can still see it was in that general area. But these snakes are just awesome climbers, and so it's just cool to see photos of that. This shot I thought was just a great shot of a cool organism. This is a moth that you can't see its wings because they're beating so fast, like a hummingbird. And what's neat about capturing photos of plants that are visiting, of insects that are visiting plants, is that you've captured an interaction here. And so if you know which species it was visiting, which plant species it was visiting, you can add that as additional data associated with the observation, or you can make a separate observation if you don't know what it is. And then once the community has been able to identify it, you can add that information. So it's cool to capture interactions in photos too. And this is neat. This is a cicada just emerged from its shell. A bunch of Eastern North America is going to see this a lot more next summer when the big brood emergence happens. But there's a couple of things to note about this. One is that it's identified to genus, the genus Neotibicin. And there's a comment further down that says that these are really hard to identify to species at this life stage. So this is a stage that doesn't last very long. It sort of hangs out there until its wings harden and then it can fly away. But it's cool to capture this in this fleeting moment. And if you do happen to see something like this, if you're able to keep an eye on it for a while, or if you're able to revisit it after maybe an hour or so, trying to get more photos of it showing its sort of more hardened adult form can help for being able to associate the adult form of something to what it looks like right as it's emerging. And then there's the spotted lanternfly. So this was in the title of the talks. This is something Steve and I talked about when we first started talking about the idea for this lecture. And the spotted lanternfly, if you haven't heard of it yet, if you're in Eastern North America, you will. The spotted lanternfly is native to Asia, and it was introduced in the Philadelphia area in 2014. And since then, it has been spreading. It's been spreading from that epicenter, and we can see the progression of its spread with observations on iNaturalist. So all of the surrounding states are on alert. Some of them have already got it. It's in Maryland, it's in Virginia, it's in New Jersey, it's in New York, and everyone else is watching for it. So unfortunately, this species can be very destructive to many crops, especially orchard crops and grapes in vineyards. So this is definitely one to watch. And you can see that the point on this map is at kind of the near the western edge of its recorded distribution so far on iNaturalist. But you can see there are a couple spots. There's a spot in Maryland, a spot in Northern Virginia. This is rapidly increasing its spread. It has not yet been confirmed in Ohio, to my knowledge, but Ohio is definitely on watch. So keep your eyes peeled. This is a distinctive organism.
So Dr. Seltzer, there was a report in the middle of our BioBlitz that somebody had found a spotted lanternfly in Ohio. Is there any update on that particular one?
So in short, no. That, as far as I know, that was not confirmed. They did, though, when it popped up on iNaturalist, a team was sent out, that sort of the local authorities were notified by people, by observers on iNaturalist, basically. And I know they went out to search the area where it was reported. For that particular observation, though, there was a question about whether or not the location was accurate. Sometimes people will, maybe a family member from somewhere else sends a picture, like, do you know what this crazy bug is? And then someone posts it to iNaturalist. And in that sharing process, it may get posted with the sort of secondary location, right? So it may have, the photo itself may have originated elsewhere, but then the person who posted it used their location to post it. So as far as we can tell, it seems like that may be what happened for that particular observation, because I haven't seen any other observations from that part of Ohio or heard it confirmed. But yeah, it's always important to consider the ways that the sort of things to keep in mind before taking action based on iNaturalist data or using it in research. There are a ton of advantages, but there are also limitations to be aware of. So if you go to the taxon page on iNaturalist, you can, I sort of blew it up bigger, but you can look at the seasonality of life stage. And this is just looking at observations that have been made in the United States. And you can see that the green is observations of eggs. The orange is observations of their nymph phase, which is really distinctive. You can kind of see it at the bottom middle almost, that like black and red and white spotted bug there. And then the adults are shown in blue. So you can see in which months you would expect to see these different life stages. And I just want to give you a snapshot of what the spread looks like based on iNaturalist data. So this is showing all of the observations that were observed of spotted lanternfly as of the end of 2017. It's just in Pennsylvania. This is by the end of 2018. You can see there's a couple others that popped up. This is by the end of 2019. And this is as of the other day. So increasingly, iNaturalist is being used by various agencies that are monitoring, keeping an eye out for invasive species. There's not really a very formal system, but because there are so many people now using iNaturalist, thankfully, word usually gets around if something unexpected pops up. And we are definitely looking to improve the tools to make it more useful for the state agencies that have this kind of responsibility for detecting invasives to enable rapid response to new invaders. So I want to talk a little bit again about a couple ways that you may be interested in using iNaturalist for yourself. Steve mentioned that he's been competing with me for yard biodiversity. I'll note I live in Washington, D.C., and I have a very small yard. Gosh, I don't remember I think the entire lot, including the footprint of the house, I think is under 4,000 square feet. You can look up my project. I have some details on the size there. Anyways, I have found over 400, almost 500 species now. There are still many species that have not been identified to species yet, so they're not included in that count. And that also does include some of my cultivated plants, because although the main purpose of iNaturalist, the main focus is really on the wild biodiversity, for keeping track of phenology, I do record usually the first bloom or the first fruit of some of the things that I have planted in my yard, which is mostly planted with native species. So something like this may be of interest to you for recording what you can find in your yard or your neighborhood or a favorite park or something like that. And as we go into winter, I really want to highlight this cool project, Never Home Alone, The Wildlife of Homes, because this is all about the probably unexpected things that you find living in your home. So if I find a stink bug, I know the Oxford area probably has a lot of brown marmorated stink bugs as well. That is, you can see, the second most observed species in this project. Also, the first is the long-bodied cellar spider, which odds are pretty good that you've got one of those somewhere in your house, as well as the house centipedes, the Asian lady beetles, and the varied carpet beetles. So the next time you find a bug in your house, if you can bring yourself to take a picture of it before you put it outside, I encourage you to post it to iNaturalist, but then also join this project and specifically add that particular observation to this project. So in addition to all of the ways that iNaturalist data is used directly from iNaturalist, we also share data with other data partners. And the biggest one of those is the Global Biodiversity Information Facility. It's also known as GBIF for short. GBIF is an aggregator of biodiversity occurrence records of many different kinds, including millions of museum specimens. So you can think of iNaturalist as kind of a virtual museum that's captured, instead of physical specimens, these digital specimens, and is sharing data with GBIF in the same way that the Smithsonian does with its natural history collection, or any of the other big international or national or even regional natural history museums that have collections. So one of the really cool things about sharing a subset of data from iNaturalist with GBIF is that it enables us to get a greater insight into the kinds of research that people are doing based on iNaturalist data. So if you...
Dr. Seltzer, Alfredo is interested to know, how can you tell the difference between a species that's expanding from a small area versus just having more people reporting it on iNaturalist, like you're showing here with the GBIF?
Yeah, that's a great question. So, so there's a couple ways you could look at that. One is you can look at the overall activity on iNaturalist in that area, and you could look at the overall activity for insects, perhaps, and you could see, first of all, I'll say iNaturalist is approximately doubling, roughly doubling every year for the last several years. So part of the growth in the number of spotted lanternfly observations is definitely due to more people using iNaturalist. However, there's also, you can also look at observations of a particular species as a proportion of the overall observations to try and control for that increase in the user base. And I think that's the main way that's been done.
Thank you.
So if you look at this, if you go to gbif.org and you search for the iNaturalist data set, and you're really interested in the other kinds of questions and other kinds of research that people are doing with iNaturalist data, this number of citations that have circled in the upper right there. If you click on that, that will link you to a page that has links to all of the publications that are using data from iNaturalist to answer some kind of question. So if you're interested in knowing more about what happens with iNaturalist data, I absolutely encourage you to check that out. It's been very cool for us to see all of the different ways that it gets used, and I think that this is a real way that sharing data adds value. So any museum that's sharing data with GBIF gets this insight into how that data is being used in a way that they probably wouldn't get if they didn't have this data sharing relationship. So I want to emphasize again that iNaturalist works because of curious people like you who share what they see and share their expertise. And so I'm excited now to turn the conversation over to Dr. Michael Moore, who's going to go into some greater depth about how he has used observations from iNaturalist to advance some really interesting questions about dragonflies.
All right, thank you, Carrie. And thanks to everyone with the Hefner Museum for giving me the invitation to chat with you tonight about some of the stuff I've been working on with iNaturalist data. It's such a pleasure to get invited to talk to an audience of non-scientists about some of the really cool research applications that we can use iNaturalist for. And so today I'm going to talk to you briefly about how your iNaturalist observations are supercharging how we study evolutionary biology. So dating all the way back to the mid-1800s, the search to understand how organisms adapt and evolve really typically and historically required many years of study. So if you take, for example, Charles Darwin, beginning with his famous voyage on the Beagle, Darwin spent more than 5 years sailing around the world, collecting specimens and contemplating what he called that mystery of mysteries, the origin and maintenance of biodiversity. But then what most people don't know is that after Darwin returned to England, he spent another 20 years becoming an expert in barnacles and setting up little experiments in the back of his British estates until he had finally fleshed out the evolution the theory of evolution by natural selection almost 30 years after he initially set sail. In addition to Darwin, Darwin's lesser-known contemporary, Alfred Russel Wallace, independently arrived at exactly the same idea at about the same time as Darwin. And also, just like Darwin, Wallace required quite a long time to really sort of formalize his ideas around the theory of evolution by natural selection. And this included a 4-year stint on the Rio Negro in South America, collecting specimens for museums, not unlike the Hefner Museum, as well as private collectors. And then after he returned to England, he immediately turned right back around and went on another 8-year collecting stint in Southeast Asia. Now, as evolutionary biologists, though, times have changed since the times of Darwin and Wallace. And whereas Darwin and Wallace needed decades to collect enough high-quality data to address those fundamental questions about the origins of biodiversity, projects like iNaturalist, eBird, GBIF, and the IUCN are giving biologists like me access to tens of millions of observations of plants and animals from all over the world, really as fast as someone can download them to their computers. And so tonight I'm going to talk to you about how citizen science is offering new ways to gain deep understanding into this mystery of mysteries, the origin and maintenance of biological diversity. Now, of course, citizen science projects like iNaturalist will never completely replace every element of the research process, but tonight I want to talk to you about 3 important ways that iNaturalist is sort of supercharging my own research, as well as some of the research of my colleagues. First, I'm going to talk to you about the role that iNaturalist can play in sort of inspiration. Then I'm going to talk to you about how important citizen science like iNaturalist can be in sort of replication. And finally, I'm going to talk to you about its sort of the capacity of citizen science projects like iNaturalist to shape important conservation implementations. So let's first begin by talking about the role of citizen science in sort of inspiring research ideas. And to do this, I'm going to tell you a story about my own research. So the hero of this story is this blue dasher dragonfly, Pachydipplex longipennis. And this is a medium-sized dragonfly. It's quite common. Probably most or all of you have seen it at some point. And it has a pretty broad distribution across much of the middle of North America. So really encompassing most of the United States, southern Canada, and down into northern Mexico. And so males of this species in particular produce this dark brown pigmentation on their wings. And this is actually sort of why I started studying them in the first place. And my work and that of others indicates that male dragonflies use this dark brown coloration on their wings to attract mates and intimidate rivals, like this really gorgeous iNaturalist picture of these two male blue dashers fighting. And so because this trait is involved in mating in this way, biologists will call it an ornament. And so you'll hear me describe this brown pigmentation on their wings as an ornament. Now, in eastern and northern North America, males almost always get a little bit of this brown wing coloration. To be sure, it can vary quite a bit among males, but they almost always get at least a little bit. However, when I started doing research on this species, I quickly became aware that field guides and reports from sort of odonate enthusiasts note that males in sort of the Western United States almost never produce any of this coloration at all. And so this got me thinking, why would this trait that's so important for a male's ability to reproduce, why would males sort of lose it in some parts of their range? So one winter, while I was pondering this question and waiting for the snow to melt, first at my home in my parents' home in New Hampshire, and then after I returned to Cleveland, I got to thinking about, well, what info would I actually need in order to explore this kind of alleged geographic pattern in these wing color ornaments of blue dashers? And so obviously I knew I would need observations of dragonfly wings. And I'd also need the geographic locations of all of those observations, sort of the GPS coordinates. Now, around this time, some folks had told me about iNaturalist, and I knew that it had pictures of dragonflies as well as the geographic locations for all of those pictures. But I assumed maybe it would have 100 observations or so. But to my great surprise, and to my continuing surprise, when I actually went to look for observations of blue dashers on iNaturalist, at the time there were about 10,000 observations. And today you can see it's almost tripled to about 30,000 observations of just this one dragonfly species. And I couldn't believe it. And in some regards, I still can't believe it. And so when I started looking at the individual observations of these dragonflies, I knew that these observations would have all of the things I would need to decisively answer this important question. And so this question that I really wanted to address essentially boils down to this. Where do males usually get a lot of this wing coloration? And where do males typically lack most of this wing coloration? And so I sorted through about 600 observations of dragonflies on iNaturalist and simply assessed whether the males in each of those observations had wing coloration across the whole sort of range of their wings that looks something like this, or whether they had little to no wing coloration, which looks something like this. Once I'd recorded all of these observations, as well as the geographic locations, I was able to map them out, and the pattern immediately became pretty obvious. So here you can see all of these circles. Those are individual observations of blue dashers. Just from iNaturalist. The circles that are shown in black are the males that had sort of more of this ornamental wing coloration. And the males that are shown in these gray circles are those that had sort of less of this ornamental coloration. And so what you can see pretty clearly is that in the northern and sort of eastern portions of the species range, males almost always have at least a little bit of this ornamental wing coloration. But as you go south and as you go west, what you tend to find are more and more males that lack this ornamental wing coloration altogether. And so the picture that this paints is that males frequently lack this ornament in the hottest parts of their geographic range. But why then would blue dashers lack this important mating trait in the hottest parts of their range? Well, the iNaturalist data that I had collected really inspired me to come up with a hypothesis, the central component of research. And so most of you are probably aware that dark coloration tends to absorb more light than paler coloration. And in animals, this additional light absorption gets converted into energy that subsequently gets converted into body heat. And so what I hypothesized was that perhaps in warm conditions like those in the American Southwest and deep into the American Southeast, that the extra heating that could be caused by the physical properties of this wing coloration might actually ultimately cause these dragonflies to overheat. And so the dragonflies might evolve to lose that coloration so that they don't overheat. So to start to test some of these ideas inspired just by these iNaturalist observations, the first thing that I needed to understand was simply whether having some of this wing coloration actually causes the males to heat up. So what I did is I captured males that had either lots of wing coloration, like this example here, or had little to no wing coloration, like this example here. And I simply heated them up and compared how their body temperatures changed through time. And so here, this black line shows the average heating rate of the males that had a lot of coloration, like this one. And this beige line shows the average heating rate of the males that had less of this coloration. And what we find pretty quickly is that the males with more of this coloration definitely reached a higher temperature when they're out there exposed to light. And in fact, what we find is that having more of this wing coloration causes those males to heat up by an additional 1 to 2 degrees Celsius. But that got me wondering, sort of, what does an extra 1 to 2 degrees Celsius actually mean for these dragonflies? And so I needed to know how much heating these dragonflies could handle. So to assess this, what I did is I measured the relationship between a dragonfly's body temperature and how well it's able to fly. And so when I made these measurements of a dragonfly's ability to fly at all of these different body temperatures, I find this sort of classic parabolic relationship whereas sort of at cold temperatures, increasing a little bit in body temperature is going to help an individual dragonfly fly better up to some sort of intermediate temperatures. But beyond those intermediate temperatures, any increases in temperature are going to make the dragonflies really bad at flying. And so based on this relationship that we estimated for a population of these dragonflies, we could then sort of calculate how much 1 to 2 degrees of extra heating is going to affect the dragonflies. And so at cool conditions, what we find is that the heating that's caused by this wing coloration is going to heat up the dragonflies, obviously, and it's going to improve their ability to fly a small amount, only about 8%. But at warm temperatures, like those that are found in the American Southwest, a 1 to 2 degree increase in body temperature that we know is caused by that wing coloration is really going to harm these dragonflies' ability to fly. It's going to cause them to overheat and cripple sort of their ability to do the things that dragonflies need to do. And so what this means is that the heating that's caused by wing coloration can in fact actually be quite dangerous to these dragonflies under warm conditions, like those observed in the hottest parts of the species range. And so from these data together, we had a pretty good potential explanation for why males lack wing coloration in the hottest parts of the species range. And that's because that extra color is going to likely lead to really dangerous levels of overheating for these dragonflies. And so what I think is so exciting about these results is not just that, it's not just sort of the biological pattern, but that the iNaturalist data is really what served as the impetus for me to design experiments that ultimately discovered a new way that these blue dasher dragonflies and maybe other animals are interacting with their environments. And so what this first story shows you, I hope, is that one important way that your iNaturalist observations are supercharging the study of adaptation is by providing inspiration for more experiments. But of course, while this story in blue dashers I think is really cool, and I hope that you all think is really cool, it's important to know whether this kind of phenomenon is occurring only in blue dashers, or whether it's a more general sort of thing that's happening to dragonflies all across North America. And this brings me to the second way that your iNaturalist observations are supercharging how biologists like me study adaptation, and that's sort of replication. Can we use iNaturalist observations to see if sort of the geographic patterns that we see in some species are more general to other species as well? So if the risk of overheating has caused blue dashers to produce less wing coloration in hot climates, then it may also cause differences in other dragonflies as well. So to test this pattern and see whether the things that we find in the blue dashers are sort of more general to other species, we used a novel technique that was developed by a group in England, also using iNaturalist observations, that allows us to measure how much of this ornamentation these dragonflies have on their wings, again, just from these iNaturalist observations. And so along with a team of undergraduates at Washington University, we measured the size of these ornaments across 2,700 different dragonflies across 10 different species. And we found something that I think is a little unexpected, but really quite exciting. So in some species, when we compare populations in the coldest parts of the species range to warmer parts of the species range, we find massive decreases in the amount of this wing coloration that they get in the warm areas, just like we find with the blue dashers. In other species, we find minor, but still what we refer to as statistically significant decreases in the warm areas of the species ranges. And finally, some of the coolest patterns that we find are actually sometimes we see that the relationship totally goes in the opposite direction, where males actually produce somewhat larger ornaments in the warmer parts of species ranges. But when we look across the entire data set, what we find is that the pattern does seem to be pretty general, where dragonflies in warmer areas produce smaller ornaments in about 80% of the species that we looked at. And so as in the blue dashers, what we find is this really remarkable pattern where populations in warmer areas typically have smaller ornaments sort of on average. So within species, we're finding that populations in warm climates have less ornamentation, and this is just using iNaturalist data. And this then got me wondering, sort of if the risk of overheating is causing differences within species, then perhaps it's also causing differences between species as well. So should we also see that species in warm climates also just tend to have less ornamentation overall? So what I did is using iNaturalist observations from the United States, Canada, and northern Mexico, I created a database of the ornamental wing coloration of all 320 North American dragonflies. Then using another sort of freely available source of data that Carrie talked about, these GBIF data, I was able to actually estimate the ranges for all 320 of those species. Then using the known sort of evolutionary tree of dragonflies, I could assess whether the species that live in the warmer parts of North America are less likely to have wing coloration than species that live in cooler parts. And so we find a pattern that looks something like this. So the species that live in warmer climates are less likely to have wing color ornaments than those species that they're most closely related to that live in cooler climates. And so just using the observations from iNaturalist and GBIF, what we've been able to document is a really remarkable pattern in evolutionary biology. What we're finding is that dragonfly populations and dragonfly species predictably adapt to warmer climates by producing less ornamental wing coloration. In addition to this being a really exciting pattern and a really interesting finding with respect to how organisms adapt to living in particular environments, I think an important takeaway for this is is that iNaturalist observations and citizen science observations can be used to test for these really general patterns in biological diversity. And specifically, this really incredible trove of observations has allowed us to test hypotheses about the predictability and consistency of evolution, a major question in biology. And so hopefully what this second story shows you is another valuable contribution that your iNaturalist observations are making to the study of adaptation is by allowing us to sort of test if patterns in one species are replicated within and among others. And so the final thing that I want to talk to you about is this sort of this last way that I see that iNaturalist observations can aid sort of our understanding of adaptation. And that's sort of by providing data on how organisms respond to environments in real time, and hopefully by giving us some information on what the best conservation strategies might be. So what my research with the iNaturalist observation shows is that dragonflies adapt to warmer climates by producing less wing coloration. And this raises the important question of what will happen to these dragonflies as the climate warms over the next 50 to 100 years. Based on the research that I've shown you, what we might expect is that the risk of overheating should cause dragonfly wing color ornaments to shrink as the planet warms. So what I wanted to know is whether we might be able to use iNaturalist data to sort of track changes in wing coloration with sort of changes in environmental temperature in real time. And so to do this, I started by gathering data from the National Weather Service, which reports how much sort of warmer or colder each year is relative to the long-term average in North America. We call this relationship the mean annual temperature anomaly for North America, which is a terrible name for a variable, but it has a very simple explanation, which is that a value of 1.1 for a year means that that year was 1.1 degrees warmer than the long-term North American average, dating back to about 1880. So values that are, so years where you see sort of a larger temperature anomaly are relatively warmer, and years where you see a smaller temperature anomaly or a negative temperature anomaly are more average or sort of less cold. And so when we take these data across years, and then we look at the ornamentation of those 10 dragonfly species that I showed you earlier, what we find is that there are changes in the average size of the dragonfly ornaments across years. And these changes in the size of the ornaments correspond exactly to where we see changes in temperature. So for example, in the 3 relative to the years either before or after, shown here in pink, what we find is that in those relatively hot years, the average size of these dragonfly ornaments declines quite a bit. And so what this indicates is that just as we predicted, ornaments do seem to shrink in response to global warming. This is potentially an important way that these dragonflies might respond as the planet continues to warm. And what I would really want everyone to take away from this is that iNaturalist observations are potentially able to provide real-time data, in this case showing that wing color ornaments are in fact smaller in warmer years. And so using iNaturalist for real-time monitoring, like I just showed you, could be a really important way to help us understand how organisms and their traits are going to respond to environmental change, and therefore are going to be useful in terms of dictating the best strategies for conserving these organisms. And so this is how I see a sort of a third crucial way that iNaturalist observations are supercharging how we study how plants and animals adapt to the environments that they live in. And so as we start to think about what research biologists like me need to be doing in the coming decades, I think that it's really clear that we live in an age where we need to gain an understanding of the origins and maintenance of biological diversity faster than ever before if we want to have any chance at preserving it. And whereas Darwin was able to spend almost 30 years gathering data and generating really deep insight into these questions, our world is changing very quickly. And we don't have the kind of time to devote to studying these questions like Darwin did before it's going to be too late. But fortunately, my opinion at least, is that citizen science projects like iNaturalist have the potential to give us data at a speed and scale that was previously unimaginable as recently even as 10 years ago. And my hope is that this will give researchers like me a fighting chance. But of course, all of this requires that we all do our part in supporting and participating in these citizen science projects. And so as I conclude here tonight, I'd like to encourage all of you to keep observing, uploading, and just generally being curious. Your work is already really helping to make major advances in our understanding of how plants and animals adapt to their environments. And you never know what kinds of things your observations might help someone else discover, or what it might cause you to discover as well. And so with that, I'd like to thank all of you for listening to me yammer on here for 20 minutes. And I think Carrie and I would be happy to take questions now.
Well, Michael, we've had a couple of a good number of good questions as you've been talking, some of which were asked just before you answered the question, and others of which are still to be asked. And maybe I'll lump some of the biology ones together right off the bat. So you've talked about the fact that wing coloration can change. Tell us a little bit more about the life history of blue dashers or dragonflies in general, how long the nymphs live, how long the adults live, when the melanin develops, and is that melanin fixed? And then we have some other questions after that.
Yeah, okay, so there's a lot to unpack there. So a basic overview of dragonfly life cycles is dragonflies have life cycles just like frogs do. So they start out in the water in sort of what we call a nymph or larval stage. And then they undergo a metamorphosis and they become these winged adults that most of you are familiar with seeing. Now, different species take different lengths of time for them to complete this life cycle. Some species can complete that life cycle in as short as a few months, whereas other species take upwards of 5 years in some cases. The blue dashers that I study throughout most of their range take about a whole year to complete their life cycle. So we tend to see them, for example, where you are in Southwest Ohio and where I am here in Missouri, we see them starting to fly around in May, and then we tend to see the last ones flying usually sometime in September. I think the last time that I collected them here was like in the middle of September. They'll mate and they'll lay their eggs into the water. The eggs will hatch that fall and they'll develop pretty rapidly before they undergo sort of an overwintering stage. And then that gets them to the final couple sort of stages of larval development. After they overwinter, sort of in the spring, once they wake up and the water temperatures are nice and warm, they grow and develop very rapidly, and then undergo sort of that metamorphosis that I mentioned before. So that's an overview of the dragonfly life cycle. The wing coloration begins to develop during the larval stage. We don't yet know a lot about the development of that wing coloration, but we do know that the things that occur in the larval stage affect how much wing coloration they will have as adults. Once they've emerged, so once they've undergone that metamorphosis and become an adult, they start to produce that wing coloration on the very first day, and it will continue to be sort of laid down, that melanin coloration will be continued to be laid down on the wings for usually up to about 2 weeks, at which point the size of the wing coloration is more or less fixed. It's not going to get any bigger or smaller after about a week or two. And so that's kind of a very brief overview of how the melanin works, in addition to how dragonfly life cycles sort of go.
So the melanin is deposited for 2 weeks in the adult wing?
Yes.
And then the adult itself lives for 30 days-ish?
Yeah, that's about right, yeah.
Okay. And so it sounds like you don't know exactly, but presumably we could take a nymph from a Minnesota lake and transplant it to New Mexico. And if you did it at the right time in its life, it would end up with pale wings.
So this is a really good question. So this was one of the things that we wanted to know after we got that first result. And it was actually something that we did in a follow-up study that I didn't get a chance to talk about tonight. But what we actually did is we took dragonfly nymphs from Cleveland, where it's quite cold and they typically get that wing coloration. And then we raised them, we put them in these experimental sort of rearing environments. And in some of them, we raised the temperature that they developed at, and other ones we left at sort of the typical temperature. And what you would expect to find if there were these sort of developmental effects like you were just talking about, is if you raise the Cleveland dragonflies at a temperature that's consistent with, say, Fresno, California, you might expect that they would not produce any of that wing coloration. But actually what we find is that when we raise them at a warmer temperature, if anything, they actually produce a little bit more wing coloration. So the developmental effects are actually in the complete opposite direction to what we find for the geographic pattern. So that strongly indicates that the patterns that we see geographically are not regulated by these kind of environmental effects during development, and suggests instead that what we're detecting are these strong sort of this strong genetic differentiation between the populations, if that makes some sense.
So as Dr. Gorchov asks, so that year-to-year change that you're talking about, the year-to-year change in ornamentation, suggests that this might be a developmental response to temperature rather than evolutionary to adaptation. But now it sounds like maybe you're saying something a little bit different.
So the year-to-year changes, based on what we showed developmentally, the year-to-year changes don't seem to be that the dragonflies know what temperature it's going to be and produce the right amount of wing coloration. It seems to be that they're either evolving very rapidly or something else is going on.
So you think this might be genetic also?
I think it might. I think it's almost entirely genetic. Yes.
And do you think that much like reptiles can be affected, the genetics of reptiles can be affected by temperature? Say, too hot makes males or females. Is any of that kind of effect happening with dragonflies?
In terms of like what sex they're going to be?
Well, or the ornamentation in this case.
Well, so we do know that it. I'm sure that I don't want to say that there's no developmental effects. What we've shown is at least the developmental effects of temperature are opposite to what you would expect. So based just on the geographic pattern, I don't know how that plays in to the geographic pattern, but it would seem, I'm sure that there's some combination of both, as there is for most traits. But our best estimate right now is that the geographic patterns and even the temporal patterns are probably related to sort of rapid evolutionary change.
Okay. Let's switch to Carrie for just a second. Then I'd like to come back to people have a handful of other dragonfly questions. But Carrie, can you tell us about the demographics of iNaturalist and any kinds of growth patterns or ways that we can contribute to the value of this data that we contribute?
Yeah, sure. So as far as the demographics of iNaturalist go, we have very little information about iNaturalist users because the only thing that we collect is your email address, and that is not shared. What we really know about people is where have you made observations. What we can say is that, I mentioned that iNaturalist is approximately doubling each year. It's growing faster in some parts of the world than others. I would say probably the most rapid growth is happening in some parts of Europe right now, I think. About a month ago, iNaturalist, when we passed 50 million observations, we did a blog post that dug into some of the geographic trends. I think that the fastest growing country in the last year has been Finland. Russia has also been growing really rapidly in terms of observation activity. I encourage everyone to go to the iNaturalist blog and look for that 50 million blog post to dig into that a little bit more.
I think one of the great things about iNaturalist using, being able to use smartphones is that that means people throughout the world largely have access. At this point, smartphones are pretty broadly accessible.
Yeah, actually, just one thing I'll add to that is that iNaturalist has been translated into dozens of languages, the mobile apps, the website, and that's all thanks to volunteers. Our translations are crowdsourced, just like all of the observations are. And so there are dozens of dedicated translators who have helped make iNaturalist accessible in other languages.
So if we have any linguists here who would like to volunteer for that, shall they just contact you?
Sure.
Wonderful. And a question that crosses between Dr. Seltzer and Dr. Moore that was asked is, how do we deal with sort of the photo sorting aspect of using the data? Dr. Moore has given a great example of looking at thousands of photographs of a single species that are going to be taken at all sorts of different angles and need to have that pigmentation mapped. Can either of you contribute some ideas as to how that's done logistically?
That's a question for Mike. I'll let him answer.
So this was a concern that actually I had when I initially did that, after I initially had done that first study. So in the first study, all I did was look at the wings, and it's very easy to tell from looking at these clear dragonfly wings if they have some pigment in them, sort of in a sense, just like yes or no. And actually, in conversations with sort of the professors that were helping with my PhD, they wanted to know whether it was possible to measure the size of the patch of the wing coloration. And I was initially quite skeptical of this. But shortly after I finished that work and finished my dissertation, this paper came out from this research project came out from this group in England, and they actually showed that you can measure the size of the patch quite reliably, as long as what you're measuring is the proportion. And in that case, all you need is sort of the relative size of the patch compared to the size of the wing. And they did these measurements at all sorts of different angles and showed that the pictures that you get from iNaturalist and these measurements that you're able to take from iNaturalist observations are almost as good as if we were to go out and catch some dragonflies and take them in really standardized conditions, as long as what you're measuring is just sort of the percentage of the wing that has that coloration on it. Now, there's other elements that as of yet we don't have a good handle on how we could measure, things associated with sort of how saturated the patch is, or how, sort of how dark it is. So there's still some limitations in that regard, but at least with respect to some of the things that I'm interested in, there are usually pretty good workarounds that folks are doing a really good job showing are just as good as if I was to take really standardized pictures of these dragonflies and measure them from those observations.
So our take-home message here is that a snapshot taken by anybody on any device should be uploaded to iNaturalist with at least the city and state, if not more precise GPS locations. But using the iNaturalist program, you can zoom in on that map and make the most precise estimate of where you were that you can think of, and that scientists like you can then use these in a productive way. Now, that segues, though, into a question about observer bias. Is it possible that more pigmented dragonflies simply avoid flying at hot times of day, or that that observers taking pictures are going out at a time of year that's going to bias one kind of coloration versus another being recorded more frequently?
That is always a concern. If dragonflies are being, I'm less concerned. So we've looked at whether you see relationships between the amount of pigmentation that you see like across a year, for example. So if it's a situation where you tend to see observers are taking fewer pigtures of dragonflies in hotter times of the year, you would expect to see that there would just be fewer observations of those dragonflies in the hottest times of year. And you might also imagine that you would find fewer dragonflies with a lot of pigmentation during the hottest time of the year. And what we actually find is that you don't see those patterns within a year. So you see differences across years, and you see differences across sort of geographic space, but you don't see sort of those seasonal differences that you might expect if there was a big observer bias associated with that, if that kind of makes some sense.
Excellent. So you've been able to control for observer bias, and in a given location, pale dragonflies are going to be there any time of day and year round, or during the flight season.
To the best of our ability to control for it so far, that's what it seems like is the case.
A couple other questions focus on some of the evolutionary or conservation aspects of this, that if we're seeing changes that potentially are being caused by something like climate change, is this going to drive dragonflies into some kind of evolutionary change? And if they're using that pigmentation in combat or wooing or something like that, is that then also going to drive change? And so what, if so, in any of that, what would you propose as a conservation method?
So I think that we probably will see evolutionary change. It's unclear how much we've seen so far. It looks like there's something going on, but in the next 50 to 100 years, we need to see what that might be. But I think, in terms of a conservation implication, and something that I would suggest is it's really important to think about the sorts of habitats that these dragonflies are going to need in order to persist. And if these traits that are really important for males' abilities to sort of attract females go away, we don't know what that's going to do to the dragonflies and their ability to persist as a species, or even the populations at the more local level. And so I think that understanding those relationships, and iNaturalist and other citizen science projects are going to be a big part of that, but understanding getting the more mechanistic understanding now of what's going to happen to those direct interactions between males and females when the males no longer have the traits that the females used to like. I think we need to study that, and then we need to use what we've learned about the geographic patterns from the iNaturalist data to prescribe places that we need to do a better job of preserving wetlands, for example. As the temperature changes, we have really nice predictions for sort of geographic change in temperature over the next 50 to 100 years. And so we can hopefully get out ahead of some of those changes and start to manage those wetland areas a little better to help preserve dragonflies and some of the other organisms that rely on those environments.
Sure. So your work, it sounds like, has really created a foundation for us to understand the kinds of questions we need to be asking, and also maybe geographically focus our efforts. And of course, you say wetlands will conserve dragonflies. And of course, if my backyard is any indication, the wetlands are going to conserve hundreds, if not thousands of species, because my backyard's not even a wetland and it has hundreds of species. Several people, well, maybe I'll go to this first and then one other. So specifically, if females prefer males with darker wings, is that true in both warmer and cooler places? So is it possible to import a male and he's going to woo all the females?
It's a great question. That was one of the things that I actually was going to be working on here at Washington University before the pandemic hit, and then we were all stuck inside. So it's still a big unknown. The dragonflies in the hottest parts of the range seem to be able to figure out how to reproduce, even though they've lost this important trait. We don't yet know how that works. And hopefully someday when the pandemic ends, I'll have a better answer to that. But it's an important, the things that the dragonflies may have figured out how to do over the course of the previous 20,000 years might not necessarily be the same as might not necessarily be the same over the next 50 years, if that makes sense. And so we can't necessarily extrapolate, how the dragonflies have figured out how to mate in Arizona to how they're going to need to figure out how to mate in 50 years in Southern Ohio, for example.
I see. So several people are asking then, and maybe a related question is, do you have any idea how this might impact other winged species or other species that have pigment changes?
So there are now there's now some people that are working on similar questions in damselflies, which are, we think of them as a closely related group to dragonflies, but actually they split off evolutionarily before there were birds. I guess they're like technically closely related, but like in the grand scheme of things, they're not that closely related. But what we're finding is that in damselflies, you actually see fairly similar patterns to what I showed you tonight in dragonflies. We've actually started to see similar kinds of patterns with breeding plumage in birds. That's some work by some folks at another university. So it seems like this interaction between temperature and these kind of mating related traits might be a really important sort of constraint on how organisms coordinate reproduction.
Interesting. So a couple of people are asking sort of related questions about separating the genetic from the environmental components. And have you done things such as transplanting late stage nymphs to environments with different temperatures to see how the adults respond? Or indeed, do females preferentially mate with those darker adults?
We haven't done a geographic sort of manipulation that way, where we take them from Minnesota and rear them in Texas and vice versa. Like I mentioned, we have taken dragonflies from cold populations and raised them at temperatures that are consistent with the temperatures that you would find in Texas. And you don't see the patterns in development that you see geographically. So the dragonflies that are reared at warmer temperatures, that grow up under warmer conditions, don't develop less wing coloration. So we don't see it that way. But one of my great dreams is to do the geographic sort of what we call a reciprocal transplant experiment, where we take the populations and swap them. I haven't done that yet, so we haven't totally sorted that out. Now, in terms of what the females might be preferring, that's a really interesting question. And as I mentioned, the females are obviously still able to choose the males that they want to mate with. In the hottest parts of these species ranges, where they have less or sometimes no wing coloration. So they figured something out to choose on, but we don't yet know what it is. Maybe the females do prefer males with less wing coloration there. There's mathematical theory that suggests that might be the case, but at this point we still don't quite know.
So we don't know if there's sexual selection. We also don't know if there's pheromones. We're just seeing directions that need study.
That's right. In terms of the hottest parts of the range, we know that in cooler parts where they have the wing coloration, females prefer it, and males are scared by other males that have more wing coloration than them.
Cool. Switching back over to Carrie for a moment, we've seen that photos can help work, such as Dr. Moore's, but how many photos should we really be taking? In your experience, how many people are demanding data sets with lots and lots of pictures? Is one enough? You talked in your presentation about having maybe host plants and the thing that's feeding on it or something. Tell us more about how many pictures we should take and how we should consider our data submissions.
So first of all, I want to address a question that I just saw come up in chat about having multiple photos associated with a single observation. And yes, that is really, really helpful. To use mushrooms as an example, you always want to try and get 3 views of a mushroom. You want to get the mushroom from the top down, you want to get the mushroom from the side, and you want to get a shot of the underside. You want to get a shot of the gills or the pores. So in general, when you approach an organism, photographing an organism for iNaturalist, you want to try and document all of its major characteristics and all of its major views to the extent you can. Sometimes it gets away. Sometimes you're looking at a little beetle and it falls off the plant and disappears into the leaf litter before you get that other shot that you really needed. So, it happens, and not all photos can be identified. Not all observations have enough documentation to be identified to species, and that's okay. Not everything gets to species. Not everything can it's cool. But you do want to try and thoroughly document. For a plant, especially something big like a tree, you want to make sure you get the bark. Ideally, you want to get a shot that shows the whole thing, because the overall shape of a tree can be really important, in addition to getting a shot of the leaves, and not just the leaves sitting on your hand or on a blank background. The leaves in context really help too. The buds, the flowers, the fruits. I mean, sometimes tree observations can really have a lot of photos associated with them. But in terms of what to look for, Steve, I mean, it can be overwhelming to be told, go out and take pictures of any wild biodiversity. So I encourage people to just start with what catches your interest. When I'm walking a trail, the sort of rules that I have in my head are blooming, moving, or dead. So I take pictures of things that are in flower, especially if it's a place where I don't know a lot of the species and I'm not going to be able to identify them very well myself. Getting pictures of the flowers helps increase the odds that someone else is going to be able to identify them. Of course, I get the flowering plant and all the rest of its features, like I just mentioned. And stuff that's moving, so anything moving catching my eye, birds, mammals, bugs, whatever, I try to take pictures of those things if I can. And dead things, because dead things don't move, and so they're much easier to photograph. I take most of my observations just with my iPhone, but I fairly recently invested in a longer lens so I can take slightly less bad photos of birds. I'm not much of a birder, but I can take a few more identifiable observations now with a better lens.
That's cool. Like I said earlier, I learned so much through the iNaturalist app every time I use it, because I'm taking pictures of things that previously I might have ignored, because it's going to take so much effort to go into the house, break out the key, try to work through it. And at the very least, the artificial intelligence on iNaturalist usually gets me to a really easy starting point. Now that said, like you're talking about with trees and things, as we start to learn things about how to identify these species, we find, oh, I need to get a picture of the leaf bud and the leaf scar and the lenticels and things. And so you can really nerd out. In fact, yesterday I took a picture of a honey locust, one of the most common trees in this region. And I just took one snapshot because, quite frankly, I was just trying to beat you with species at that point. And I was running down the street really fast. And the AI from iNaturalist said, "Is this a spider monkey?" And I looked back at the picture. I was like, you know what? It's really easy to misinterpret this picture as a spider monkey. And so I sat down and I did my scientific due diligence, and I took a handful of more useful scientific photographs, and it was fun just to have that interaction with AI too.
That's pretty funny, Steve. I'm glad you didn't report a spider monkey from Ohio.
Well, Dr. Seltzer and Dr. Moore, it's really been a pleasure to have both of you here and telling us about ways not only that our data can be collected, but then they are actually used in real science. And Dr. Moore, you've given us some great conclusions about science, but you've also shown us that here's now a foundation of questions and some directions to search. And I'm sure that we have lots of students who are watching us right now who are thinking, oh, maybe I can partner with him, or maybe I can go and do that off on my own. And that's one of the great things about science, I think, is here we see that, the three segments here, Dr. Seltzer, Dr. Moore, and our entire audience have been collaborators here to come up with not only amazing answers, but also really useful questions. I'm really thankful to all of the people who have contributed questions through all of our various channels. As I said, I'm thankful to all of our volunteers and sponsors and everyone who contributes to the museum on literally a daily basis. I think this has been a great way to host our 46th annual Hefner Lecture. And nonetheless, I hope that we'll be able to have share some food together next year and visit the museum. Unless there are any further questions, we will sign off tonight with a great thanks to our panelists, Dr. Seltzer and Dr. Moore.
Thanks so much, everyone. I hope to see you on iNaturalist.
Yes, thank you.
All right. Thanks, everyone. We'll be talking to you at the museum as soon as we can.
Thank you, everyone.